Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video, with a continuation of our lecture series on histopathology, we're going to be discussing instrumentation that is used in the histology laboratory. All right, let's get started. The first instrument in the histopathology laboratory that we are going to be discussing is the microscope. So microscopes are critically important and are used for quality control monitoring of the histology laboratory specimens. Slides prepared in the laboratory should be reviewed with the microscope to examine the quality of their processing, sectioning, and staining. There are a couple of different types of microscopes we're going to be briefly discussing in this presentation. These are light microscopes, polarizing microscopes, phase contrast microscopes, fluorescence microscopes, dark field microscopes, and lastly, electron microscopes. So, and as a note here, this is not going to be an in-depth discussion of microscopes, just an overview of the types of scopes and briefly about their care. The picture on the right-hand side of this slide shows a compound, or also called a light microscope. So this is the ocular lens, or the eyepiece. These are the objective lenses here. These come in different powers. So this is the stage. This is where your slide will be put on to look at it under the microscope. The fine and coarse adjustment knobs here. All right, that's going to help you focus on the specimen. This is the condenser and diaphragm here. This here. I realize my arrow is kind of pointing a little bit to the stage, but that's where I circled is what I'm talking about, the condenser and the diaphragm. And then this is the light source here. All right. In the histopathology laboratory, light microscopes are used to examine tissue sections on slides. So the lens system on the light microscope consists of the objective lenses and the ocular lenses, which of course are those eyepieces. So each of the objective lenses provide both magnification of the image as well as image resolution. The stage can be moved up and down and consists of the condenser and the diaphragm. The condenser helps to concentrate the light from the light source on the tissue section. Total magnification of the specimen is determined by multiplying both the magnifications of the ocular lens and the objective lens. Before we discuss the other microscope types, let's talk a bit about general light microscope care. So microscopes come with a protective cover. When not in use, this cover should be on the microscope. The lenses must be cleaned frequently with designated lens paper. Do not use any other paper tissue for this, or the lenses will get scratched. Only designated lens paper. Immersion oil is used with higher power objectives. This is oil that goes on the lens to increase the resolving power of the microscope. And it is, in fact, an oil, and this must be cleaned off immediately after use. It is also important to note that when using oil immersion, you do not want to drag other objective lenses that do not use the oil through it. This can damage those lenses. Do not ever dismantle the objective lenses either. Now, when not in use, make sure to turn off the light source of the microscope. Also, when not in use, the slide that is put up on the stage for viewing needs to be removed. The scope should be focused upwards, not downwards, especially when you're working with higher power objectives. So this means you want to start with a lower power objective and work your way up to a higher power objective. Lastly, never touch the surface of the lens. This can damage it. Microscopes are, of course, very expensive pieces of equipment and should be treated with care. Polarizing microscopes are those that use a polarizer between the light source of the microscope and the specimen, and inserts a second polarizing filter between the specimen and the viewer's eye. Natural light vibrates in various planes, whereas light from the polarizing microscope vibrates in a single plane. If the path of the light is parallel, light passes through the microscopic field of view and will appear bright. If the polarizer is rotated, crossing optical paths, the light is blocked and the microscopic field of view will appear dark. In this case, material that has the property of anisotropism or birefringence will appear lit up on this dark background. Polarizing microscopes are used primarily for the identifications of crystals like talc, silica, or urate crystals. It can also be used to make the identification of amyloid stain with Congo red stain more specific. And we will discuss this more in our carbohydrates and amyloid lecture. Light microscopes can be converted into a polarizing microscope by placing a polarizing film on top of the source of light and another on the top of the microscope slide. So this is an example of what I was just talking about. So the photo on the left-hand side of the slide here shows a kidney tissue section that is stained with Congo red stain. So this is a photo taken from a lighter compound microscope view. The orangish red stain in this photo shows amyloid. And again, we'll talk about this more during our carbohydrates and amyloid lecture, but amyloid is a fibrous protein that is deposited into tissues under certain pathological conditions. So that orangish red is that amyloid in that kidney tissue. Now on the right hand side is the same kidney tissue section, this time being viewed with a polarizing microscope. See that light green glow on that right hand side photo? So that is the amyloid, which is birefringent, so it'll light up under a polarizing scope. So on the left hand side is a light scope, on the right hand side is the polarizing scope view. Phase contrast microscopes utilize shifts in light passing through a transparent specimen to brightness changes in the image. So phase shifts themselves are invisible but become visible when shown as variations in brightness. Phase contrast microscopes are used for viewing unstained specimens. They're particularly good for unstained cells that are still alive. These are not used frequently in histopathology laboratories. Like with polarization, light microscopes can be converted to phase contrast by putting special phase contrast equipment on the microscope. Dark field microscopes only utilize scattered light, so objects appear larger using dark field microscopy, and finer structures are seen better with this type of microscope than with a light microscope. This is used most frequently for unstained microorganisms and rarely has a use in the histopathology laboratory. In fluorescent microscopes, the specimen is bombarded with short wavelength light in the ultraviolet, violet, or blue range. Some compounds like collagen fibers, as an example, just fluoresce naturally under the fluorescent microscope. This is called primary fluorescence. Those specimens that do not have that natural or primary fluorescence must interact with a fluorochrome, which is a dye that fluoresces if excited by UV light. Either an antibody, antigen, or complement is labeled with this fluorescent dye. One of these dyes is called fluorescein isothiocyanate, or FITC. This is a photo from Dr. Carson's book that shows direct immunofluorescence of the kidney glomerular basement membrane. It is coated with a goat anti-human IgG, which is an antibody. The electron microscope is the last microscope we will be briefly discussing in this lecture. Electron microscopes use a beam of electrons as a source of its illumination. There are two types of electron microscopes, transmission and scanning. In the transmission type, the specimen either transmits the electrons, which produces a clear area on the image, or deflects electrons, which produces a dark area in the image. Now, a scanning electron microscope produces a 3D image and is great to study the small surfaces of a specimen. Now, this is a microscope, so any type of electron microscope, either transmission or scanning, that is used primarily in research laboratories. So this is a photo of a kidney section from Dr. Carson's book that is being viewed using an electron microscope. All right, so that's it for microscopes. So now let's talk about microtomes. So these are instruments, as you can see on the right-hand side of the screen here, that are used for cutting very thin sections of tissues for their examination under a microscope. There are three main types of microtomes: rotary, sliding, and clinical freezing. And histopathology laboratories where paraffin, embedded, and frozen sections are the main requirements, the rotary microtome is the most commonly used type of microtome. Rotary microtomes operate with a screw feed. The tissue block that is put into the microtome moves up and down. Either the microtome knife or the tissue block advances at a preset length with each revolution of the microtome wheel. Rotary microtomes are the most commonly found type of microtome that is found within cryostats. And we'll discuss cryostats here in a few minutes within this lecture. There are manual rotary microtomes where the user has to manually rotate the wheel in order to cut the tissue block, and then there are also automated ones. The automated rotary microtomes have a motor that is often controlled with a foot pedal. In a sliding microtome, the tissue block is held stationary while the microtome knife is moved along a horizontal plane past the tissue block. The microtome knife completes its cycle and then returns to its starting position. A screw feed then causes the tissue block to be raised toward the knife at a preset length to be cut again. The sliding microtome is used for cutting colloidin and large paraffin blocks. Recall that in our processing lecture we talked about colloidin. Hopefully you remember it. This is another type of embedding medium for tissue samples. Sliding microtomes are not used routinely in the histopathology laboratory. Like with microscopes, when not in use, the microtome should be kept covered. At the end of the day, all the accumulated paraffin should be removed from the microtome with a soft brush. You can also use a soft cloth that is moistened with xylene to get off all of that paraffin. Lubricating of the microtomes should be performed on a routine schedule as designated by the microtome's manufacturer. And if the microtome requires repairing, someone that is skilled in microtome maintenance should perform that repair. It's also important to note that all service, repair, and routine preventative maintenance should of course be kept documented in the laboratory. Clinical freezing microtomes have largely been replaced by the cryostat. These are a portable tabletop microtome that clamps to the table. It has an attached supply of carbon dioxide that helps to freeze the tissue sample. The knife of the clinical freezing microtome swings out over the frozen tissue sample, and as it returns and makes a complete cycle, a micrometer screw advances the tissue block towards, upward towards the knife. Transmission of airborne disease is more likely to occur with this clinical freezing microtome because the bursts of carbon dioxide may spread human tissue. And like I said, it's mainly been replaced by the cryostat. It is important that the knife in the microtome is sharp and has an edge that is free of defects. Glass knives are used for cutting specimens that are embedded in plastic. Ralph knives are glass microtome knives that are specific for cutting sections of tissues that are embedded in glycol methacrylate. So recall in our processing lectures that glycol methacrylate is a type of plastic embedding media. Microtome knives are used for cutting paraffin, colloidin, carbowax, and frozen tissue. Disposable blades have replaced knives in many histopathology laboratories. These are special stainless steel blades. When talking about microtome blades, the bevel angle and the clearance angle are important. The quality of the tissue section is determined by the condition of the edge of the knife blade and the clearance angle. The knife of the microtome has a wedge angle of 15 to 18 degrees. A cutting facet is on each side. These cutting facets intersect at an angle which is known as the bevel angle, and this needs to be at 27 to 32 degrees. The clearance angle is the tilt of the knife in relation to the tissue block that it is cutting. This needs to be from 3 to 8 degrees. This picture shows these angles that we're referring to, so the bevel angle and the clearance angle. The quality of the tissue section is determined by the condition of the edge of the knife and the clearance angle. Again, clearance angle should be 3 to 8 degrees. There is an optimum cutting speed for each type of tissue. It's dependent on the type of material, angle of the knife, condition of the knife edge, and the thickness of the tissue section. A general guide for paraffin sections is that the wheel of the microtome should be rotated 1 complete revolution per second. For the majority of routine work, the section should not be more than 1 cell layer thick and should be set to cut at 4 to 5 microns on the micrometer scale. You can tell if you have correct thickness by viewing the section on the microscope. If a section is 4 to 5 microns in thickness, all nuclei in the sample will be in the same plane of focus under the scope. There are various things that can happen when cutting tissue sections with the microtome. Irregular, skipped, or excessively thick and thin tissue sections with the microtome are the result of too little or too much clearance angle. This can be corrected by adjusting the clearance angle between the micrometer blade and the tissue block. Sections that are grooved, scored, smeared, or deformed are usually produced by a dull edge of the knife or blade. Replacing the blade will correct for this. You can also move the existing blade to an unused surface. This will also correct this. So moving the blade to an unused area will also help if there are scratches and splits in the tissue sections. If the tissue sections are mushy, this is caused by improper dehydration or clearing of the tissue sample during the processing steps. If this happens, the tissue can be taken back into the dehydrating agent and then reprocessed. This certainly helps, but the tissue sections will not be as good as a well-processed tissue sample. The picture on the left-hand side of this slide is that bevel and clearance angle picture again, just to remind you of what they look like in reference to the tissue block. The picture on the right-hand side of the slide shows an A and B. So A shows when the clearance angle is too slight. So this causes sections that can be missed or skipped, too thick or thin, and wrinkled or jammed sections. The B section shows when the clearance angle is too great. So this causes chatter, microvibration, washboarding, and undulations. These make it difficult to get a proper ribbon, and we'll talk about these errors here momentarily. Crooked ribbons occur when the horizontal edges of the tissue block are not parallel. This can also be caused by the lower edge of the tissue block not being parallel to the edge of the knife. This can be prevented by ensuring that the upper and lower edges of the tissue block are parallel. Also, of course, that the lower edge of the tissue block is parallel to the edge of the knife that is going to cut it. You should also ensure that there are no issues with the edge of the blade and also that the tissue block is evenly chilled. If the face of the tissue block is not parallel to the blade in the microtome, one side of that tissue block will be exhausted while the user attempts to get a complete section of the tissue block face. This wastes tissue and can cause the loss of vital areas of the tissue sample. This can be prevented by making sure the tissue block holder is adjusted so that the face of the block and the blade of the microtome are parallel to each other. Photos showing this are on the next slide here. So the photo on the left-hand side shows a tissue block that was not parallel to the blade edge. The photo on the right-hand side shows an example of when the entire face of the block was not parallel to the blade of the microtome. This resulted in uneven ribboning and a major part of one side of the block that got cut away. Holes can occur in the tissue section, so this is caused by facing the tissue block too aggressively. Small flecks of tissue are removed from the block, leaving a hole. Liver tissue and tissues of the lymph node are especially prone to holes occurring from this error. So when holes are seen in the specimen and are the result of artifact from improper sectioning, the holes will decrease in size in each successive ribbon until they finally disappear. Now, holes can also appear when air in the tissue is not fully displaced during its infiltration processing. So this occurs most frequently within lung tissue. And these are the type of holes that do not disappear with continual ribboning. And the reason for that is it's not the result of the microtome, but the tissue itself. Holes in the tissue section can be prevented in a number of ways. You can soak the block briefly in ice water or with a wet piece of cotton before the sectioning process. If there is a lot of tissue, you can cut and discard the ribbons until those holes disappear. This really isn't a great method as you can potentially lose important parts of that tissue. You can face the tissue block less aggressively by making smaller micrometer advances. For large brain tissue specimens, you should cut at 5 micron intervals until the tissue is exposed, and this will help to prevent those holes. So here are two examples of holes in the tissue section. So the photo on the left-hand side of the slide here shows numerous holes in a central nervous system tissue. The photo on the right-hand side of the slide shows another example of holes in a tissue specimen. These both were caused by facing the tissue block too aggressively. So I've used the word ribbon briefly in this lecture. So the term ribbon refers to the sequence of connecting sections of cut tissue that are pulled from the microtome. And the formation of a proper ribbon depends on if there is enough heat that is being generated by the friction of each section, which in turn causes the sections to adhere to each other. Sometimes the ribbon will not form, and there's many different reasons for this, although the most common reason is because the blade of the on the microtome is too dull. It can also be caused by the paraffin wax being too sticky or too hard. There can be too much blade tilt, or the temperature of the room could be either too high or too low. These reasons can be corrected by choosing a paraffin with a lower melting point. Also, by decreasing the tilt of the blade, so creating a smaller clearance angle, or by changing the room temperature. And of course, these solutions depend on what was causing the ribbon not to form in the first place. Sometimes the sections lift from the knife as the tissue block is raised. Most frequently, this is caused by either a dull knife blade or by too little of a tilt of the knife. So this can also be caused by too warm of a room or by paraffin that is too soft. This error can be prevented by creating a greater clearance angle by increasing the knife tilt, or by changing to a harder paraffin with a slightly higher melting point. Something called washboarding or undulations can occur in tissue sections using a microtome. This occurs most frequently in very hard tissue like uterine tissue. It often occurs when a tissue block or knife hasn't been properly clamped within the microtome. It can also be caused by too much knife tilt and from worn microtome parts. This can be prevented by making sure that the paraffin is filled to the top of the cassette to provide support for the tissue. Also, ensuring that the tissue block and the blade are properly clamped into the microtome can help prevent this. The block holder shaft should also not be overextended, as this may cause undulations within the section. Decreasing the tilt of the blade, and also just making sure that the microtome is working well and is up to date on its routine maintenance can help prevent this from happening. This is an example of washboarding, also called undulations in a tissue section. So you see these see how it kind of looks like kind of crinkled or wrinkled, sort of like an accordion type of sort of wrinkling, so that's what undulations are. The photo on the left-hand side of this slide shows a block holder shaft that is overextended, so you can see how it's overextended here. All right. This can cause the vibration during tissue sectioning, which can then cause that washboarding. The right-hand side photo shows a block holder shaft that is correctly positioned. Chatter or microscopic vibration is another thing that can occur with the use of a microtome. This is most frequently caused by the tissue being over dehydrated or just having a lack of moisture within that tissue. It can also be caused by a microtome blade being too dull or by having too much blade tilt. Ensuring that the tissue does not become over dehydrated is a way to prevent this from happening. If it is over dehydrated, the tissue can be moistened by facing the block and soaking it really briefly in ice water, or by putting a moistened tissue paper on it. Also, you can decrease the tilt of the blade, so creating a smaller clearance angle. And lastly, you can also decrease the cutting speed of the tissue. And recall, for routine microtome use, 1 revolution of the wheel per second is considered to be the ideal cutting speed. This photo shows what chatter or microvibration looks like in a tissue section. Again, this most often is caused from the tissue being over dehydrated, which is an error in the processing step. So if you see here, there's like white lines that go like all the way across the tissue, right? You see those kind of, that's the chatter, the microvibration. Skip sections, or sections that have like a varied thickness, can be caused by too little of a tilt of the knife blade. It can also be caused by the parts of the microtome just being worn out. Sections that are compressed, wrinkled, or jammed can be caused by a number of things. So these include a dull knife blade, a knife blade that is gummed with paraffin wax. If the paraffin wax is sticking to the back side of the holder, that can cause that. Cutting the tissue too quickly or by having too little tilt of the blade. Also, too warm of a room can cause this to happen. So this can be prevented by wiping up excess paraffin, keeping the microtome clean, making sure that you're using a sharp knife blade, and of course increasing the tilt of the blade. The photo on the far left-hand side of this slide shows a perfect ribbon. No defects are present within this ribbon. It looks great. That should be what we're shooting for, that far left picture. The photo in the middle of the slide shows compressed sections in a ribbon, and the photo on the far right-hand side of the slide shows what a major nick in the knife edge can cause in a ribbon. Sometimes sections will fly and stick to the microtome or to nearby objects. So this is the result of static electricity. So this can be prevented by using a humidifier in the laboratory. This adds moisture to the air, reducing that static. You can also find a way of ionizing the air to prevent this from happening. All right, so that's it for microtomes. Let's start talking a little bit about the cryostat. So a cryostat is a refrigerated chamber that contains a rotary microtome. So this is a device that is used to create frozen sections. So the knife must be kept sharp, just like with other microtomes. And actually a wedge-shaped knife is most commonly used in these. So it contains something called an anti-roll plate. So this is a square rectangular piece of plastic or glass that rests against the knife blade and allows the tissue to slide between it and the knife blade. So this helps to produce flatter tissue sections and allow it to be picked up on the slide with fewer wrinkles. The tilt of the blade edge should be 30 degrees between the center line of the knife and the face of the tissue block. The knife should be tilted more for frozen sections in comparison to paraffin wax embedded tissues. So most cryostats should be operated at around -20 degrees Celsius. Now some tissues require slightly higher temperatures, so these include tissues from the brain, liver, lymph nodes, spleen, and the endometrium. So here's a photo of a cryostat. This particular one comes from a brand called Leica. Here's a photo showing that anti-roll plate that I was talking about. So recall, this is in a cryostat, and this rests against the knife blade and allows the tissue to slide between it and the knife blade. This helps to produce flatter tissue sections and allow it to be picked up on the slide with fewer wrinkles present. If the tissue is slowly frozen, this allows the water that is present in that tissue to start to form ice crystals. And the size of these ice crystals is proportional to the speed in which it freezes. These ice crystals cause holes when viewing the sections under the microscope. Tissues can be frozen quicker by using either a heat extractor attachment, dry ice, Freon spray, or isopentane that is suspended in liquid nitrogen. This helps to prevent this ice crystal formation from happening. For skeletal muscle tissue biopsy specimens, cooling by the isopentane that's suspended in liquid nitrogen is the preferred method. A tissue processor is a piece of equipment that automatically processes tissues. There are two main types of tissue processors, an open system or tissue transfer, which transports tissues from one solution to the next solution, and a closed system or fluid transfer, where the tissue is held still and the necessary fluids are pumped in and out of it. The closed system is completely computerized and prevents specimens from drying out. One disadvantage of this is not all reagent types that are necessary for tissue processing can be used on these processors. There are also microwave processors and help with better turnaround times. It helps to speed up the decalcification process. There are two types of automatic stainers, linear and robotic stainers. Linear automatic stainers transfer the slides from one container to the next. Robotic stainers are completely computerized and allow for exact staining times. Automatic cover slippers, as seen in the photo on this slide, do what you think that they would. They take slides and apply either glass or plastic cover slips to them. These are obviously very helpful and reduce time in cover slipping if compared to doing it manually. Flotation baths are used in the histopathology laboratory to float out paraffin ribbons. Ribbons should be stretched gently on the flotation bath, and any wrinkles or folds should be gently teased out of the section. The water temperature is usually kept around 5 to 10 degrees Celsius below the melting point of the paraffin that is being used. There is an artifact called parched earth that can occur if the flotation bath water is too hot. So this photo shows an example of what parched earth looks like. Again, this may happen when the tissue has been in a flotation bath that is too hot. If paraffin sections are picked up on clean and untreated glass slides, they will likely wash off during the staining process. Because of this, an adhesive is generally used. These adhesives are chromium potassium sulfate, poly-L-lysine, and amino-alkylsilene. So chromium potassium sulfate coated slides can be created by dissolving 1 gram of gelatin and 1 liter of distilled water and then heated. The solution must be cooled and 0.1 grams of chromium potassium sulfate must be added. The slide should be dipped several times in the solution, then drained and dried in a vertical position. Poly-L-lysine coated slides can be created by dissolving 25 milligrams of poly-L-lysine in 26 milliliters of distilled water in a small flask or a beaker. An applicator stick should be stuck down into the solution and rolled over the slide to create a coating. These slides should be dried overnight at around 50 to 55 degrees Celsius. Amino-alkylsilene treated slides can be created by soaking slides in clean acetone. They should then be air dried and then soaked for 2 minutes in a solution containing 250 milliliters of acetone and 5 milliliters of 3-aminopropyl triethioxysilane. After this, the slide should be rinsed in 2 changes of distilled water and dried at 60 degrees Celsius for around 20 minutes. Any excess of those adhesives should be removed, as leaving it will stain and make the sections appear messy. You can add 1/4 teaspoon of gelatin to the flotation bath to allow for good section adherence. Millipore filtered DI water is recommended for the flotation bath, because it helps to prevent the growth of any microorganisms in the flotation bath. After your tissue sections are mounted properly on the slide, it should then be dried in an oven or warming plate. The slides have to be completely dry before they are deparaffinized. If the paraffin is not completely removed, it will cause white spots to show up on the tissues. Dryers and ovens are usually maintained at a temperature that is just above the melting point of paraffin, so usually around 60 degrees Celsius. Incubators are maintained at around body temperature, which is 37 degrees Celsius. These are used for various enzyme reactions and some special staining applications. It is important to note that these incubators should not be used for the purpose of drying slides. Circulating water baths are also used in the histopathology laboratory. These are tabletop machines that have water whose temperature can be controlled. To get a solution to a desired temperature, the water bath is set to that temperature. Then the solution, or whatever you want heated up, is put in either a plastic or a glass Copeland jar, and that jar is covered. The jar is then placed into that water bath. The heat from the circulating water in the bath brings the solution to that desired temperature. So you can see an example of a water bath here on the right-hand side. So the temperature is controlled on this interface here. This is a power button, and then there is a lid that goes on top of the water bath that you just remove to place things in the water bath. Both freezers and refrigerators are important for the histopathology lab. We use them to store many of the reagents that we use in histology. Fridges are usually kept around 4 degrees Celsius and freezers at negative 20 degrees Celsius. There are also like deep freezers that are kept at negative 70 degrees Celsius, and these can be used for long-term storage of antibodies. pH meters, like the one pictured here on this slide, are used in the histopathology laboratory to measure the acidity of solutions. So these meters are particularly important for techniques in enzyme histochemistry because the majority of the solutions used in there are required to be at a certain pH level. Accurate weights of substances for the preparation of solutions are obtained by using a balancer scale. To properly weigh a substance, an empty weight boat must be placed on the scale. The scale must be tared or zeroed out with the weight boat still on it. The chemical must be put into the weight boat and added into the desired weight. So the weight boat and the chemical must then be removed. It is important to keep the scale clean as well as the area surrounding it clean. This is important for keeping accuracy of the scale. A tissue embedding center is a piece of equipment that provides a supply of melted paraffin wax, warm storage for embedding molds, and a warm and chilled plate for tissue blocks. A photo of a tissue embedding center is shown on the right-hand side of the screen here. Micrometer pipettes, or also called micropipettes, are a way to accurately and precisely transfer volumes of liquid in the microliter range. These have an adjustable volume from range of 1 to 1,000 microliters. These pipettes should never be taken out of their designated range. Any measurement outside of that designated range is inaccurate. Solvent recyclers are large machines that distill chemicals for their reuse. This can be used for xylene, alcohol, and xylene substitutes. It allows these chemicals to be reused multiple times if desired. All right, so that's it for our instrumentation lecture in histopathology. If this video helped you out, go ahead and give it a like, and please make sure to subscribe to my channel for more educational laboratory content. And as always, if you have any questions, feel free to leave those down in the comments below. I try to respond to every comment that I get. All right, until next time.